So Huntington's disease is a monogenic neurodegenerative disease. It is, in fact, I think the first one for which the causative gene was identified. It is caused by a trinucleotide expansion in the Huntington gene, and the specific trinucleotide that expands is CAG. So there is an expansion of this. The gene has a number of CAGs that encode for glutamine, and in people with Huntington's disease, there are 39 or more. And if you have more than 39 repeats, then genetics is destiny. You will get Huntington's disease at some point in your life. The more repeats you have, the earlier you get the disease. So if you have 40 or 41 or 42, you might get it in your 40s or 50s. But if you have, 47 or 48 or 49, you probably get it as a child or as a teenager. And that's particularly awful. The disease is associated with the production of both a toxic RNA. So the RNA itself is thought to be somewhat toxic. But then in addition to that, of course, there's a toxic protein. And the protein has all of these glutamines, and the glutamines somehow aggregate, and the protein aggregates accumulate. And they first accumulate in this deep brain structure called the striatum, which is in the very center of your brain and is composed of these two things, the putamen and the caudate nucleus. And this is the part of your brain that does two really important things. It controls movement, and that's why people with Huntington's disease develop these movement disorders. They can develop a kind of Parkinsonism where they don't move very easily, but they also develop these choreiform movements where they can't control their movements and have, are kind of hypermotile. And then in addition to that, they have psychiatric problems because, again, the striatum receives the dopamine inputs that are important for reward and motivation. Once over time, as the cells in the striatum degenerate, then the degeneration spreads. And that spread is at least partly due to a kind of prion-like spread. So this mutant protein somehow gets secreted and it gets carried to cells that are connected to those cells that are first degenerating, and those cells degenerate. So you start then getting degeneration of the cortex, and so people then start having these cognitive problems. The disease is, of course, devastating. It is the worst kind of combination of Parkinson's and Alzheimer's and ALS, and it progresses on average from sort of diagnosis through to death is on the order of about 10 years, but it varies a bit. So that's kind of what we know now. And maybe the most recent, the most recent idea in Huntington's disease is that we used to think that the number of repeats people had was stable, and that you have whatever 40 or 41 or 42, and that's what you're always going to have. But it turns out that in some of these cells in the brain, there is actually an increase in the number of repeats, even though these cells don't divide. Somehow they keep accumulating these CAG repeats. So there's what is called somatic expansion, and this is particularly bad in the striatum. So in the striatum, there's a lot of DNA damage, and every time that your genome gets repaired, you add some more CAGs, which means that you start making more and more toxic protein. So that's where we are in terms of the kind of science of Huntington's disease.